All right, welcome to this video series. We're covering introductory biology and AP biology. And I combine these together because they're effectively the same thing, right? Passing an AP course and the AP exam successfully is supposed to be the equivalent of a first-year sequence in biology for anyone majoring in biology or taking it as a prerequisite to medical school or any other program. And this textbook, Biology 2E by OpenStax, serves both of those purposes really well. So anyone who's taking a first-year course as a biology major or a high school class that's given for college credit, or if you're studying for the AP class, these topics should cover all of those purposes. And if you want to follow along in the OpenStax book, it's completely free of charge. You can download a PDF by just Google searching OpenStax Biology 2E. You can go along with me. I'm going to go through these PowerPoints and explain the concepts in more detail, try to relate them to other concepts in biology, use analogies to make things make sense, and the playlist will go in order. So follow the playlist, subscribe to the channel, and as I make these videos and go through the entire book, you'll get updates all along the way. All right, so the first thing that we have to tackle in understanding biology is foundational chemistry. Now, the AP biology or your college course likely doesn't have a lot of chemistry in it, but you're going to have to understand some basics. And so this is a very, very fundamental, like first concepts primer. So once we get through this, we can move on to bio stuff. But for right now, fundamentals of chemistry. This model of an atom is showing us a few different things. This structure at the center of every atom you call a nucleus, and a nucleus contains two different particle types. Neutrons that don't have any charge at all, hence neutron, and protons that have a +1 charge, they're positive. Now, around the nucleus, you have smaller, much lighter particles called electrons that each have a negative charge that are orbiting that nucleus. And the way this works is the number of protons that you have inside of a nucleus define what an element is. So whether it's carbon or oxygen or nitrogen, if the number of protons changes, the element that you're working with has now changed. That doesn't happen biologically. That only happens in a nuclear reactor or in a star that's burning or exploding. So we don't really have to worry about it changing. The number of neutrons absolutely can change, and we'll see that later in something called isotopes. The number of electrons can also potentially change, but that will give our atom a charge. So in this example, you have two protons, you have two electrons. The protons are positively charged, the electrons are negatively charged. They all cancel out, and so the net charge of this particular atom equals 0. If we were to add an extra electron, then we would have a charge of -1 because we have 3 negative charges and 2 positives. If we were to remove these electrons so that we only had 1 left, then we would have a charge of +1, 2 protons, 1 electron. Okay, so neutrons can change in number, that gives you isotopes. Electrons can change in number, that gives you charge, which we call ions. Protons do not change unless, again, nuclear reaction means we have an entirely different element now. Okay. All right, so reading the periodic table to make sense out of these things, when you see chemical symbols, the single or 2-letter abbreviations that you see are just to simplify this so we can make the font bigger. C happens to be carbon, O is oxygen, N is nitrogen. These are the elements that you're going to deal the most with in biology, along with hydrogen, but there are many other on the table. So if you read them, you'll primarily see 2 numbers. This number is the mass number. That tells you the total mass or weight of the atom, and the atomic mass number, that tells you the number of protons present. And so carbon is defined as always having 6 protons. If you change that number, it's not carbon anymore. So in this example, if this particular isotope of carbon has 6 protons and its total mass is 12, we know that it must have 6 neutrons, right? Because protons and neutrons are the only particles with significant mass. Electrons don't really weigh much of anything. And so if you know the total's 12, protons are 6, neutrons must be 6. In this example, you know that if your total mass is 13 and your protons are 6, that you must have 7 neutrons, right? These are both carbon because they both have 6 protons, which defines an element, but the number of neutrons changes, so we say that they're different isotopes, right? They have slightly different weight. All right. You can apply this to any other element. You shouldn't have to do too much of this, but remember, when you see elements of carbon, isotopes of carbon, isotopes of nitrogen, that's all we mean. Same protons, different neutrons, so the mass changes. All right. The next part of this is realizing that electrons don't always stay in the exact same place. Right? So this Bohr model of the atom is just named after the scientist that first proposed it. And the idea is the electrons that surround the nucleus, right? For this hydrogen atom, this is our nucleus. Hydrogen only happens to have 1 proton and 1 electron. That electron can occupy different shells, we say, or orbitals. And so 1n, 2n, and 3n just represent different positions or shells that electron can be in. Now, the electron is negatively charged, the nucleus is positively charged. They're attracted to each other and they want to be close. And so all you really need to remember is when you shine any sort of electromagnetic radiation, right, which just means visible light, X-rays, microwaves, gamma rays, if you shine energy in the form of light onto this atom, that electron can be excited. Right? And since it's attracted to the nucleus, it's going to take more and more energy to push it to higher and higher levels. And so the further away from the nucleus your electron is in these shells, the higher the energy it's going to have. And then if you take the light source away, the electron can fall back down to a lower level. And in the process, it can actually emit light. Right? And you can see this, we're not going to go into it here, but you can see that light being emitted and determine what element you're looking at. That's how astronomers look at stars from, light years away and determine what they're made out of based off of what wavelengths of light are being, emitted back out from those electrons. Okay. So we use these electron shells to determine how the periodic table is set up. Right? So periodic table is set up in groups and in periods. The groups, the thing that they have in common is the filling of their electron shells, and the periods, the thing that they have in common is they're arranged by increasing size, right? And so as we go across the period, we're adding in more protons as we move across so that we get larger and larger elements. And the groups, since they have their last electron in the same shell, they have similar properties. Right, so when you actually visualize this on the periodic table, these are your periods going across. These are your groups going up and down. And for the sake of biology, all you really need to know is that these groups or columns, the elements in those columns tend to behave in similar ways. For example, hydrogen, lithium, sodium, potassium, etc., when they form ionic bonds, they all take on a +1 charge, right? The reason for that is they have 1 extra electron, right, in their next electron shell, and they kind of like getting rid of it because it makes them fall back down and look like these very stable noble gases on the other side. Okay. Same thing, calcium, magnesium, etc., they tend to take on a +2 charge. On the other side, things in this column, chlorine, bromine, iodine, they tend to take on a -1 charge because they like getting an extra electron to look like those noble gases. All right, so reading the periodic table, now that you have that basic information, you can read atomic masses, atomic numbers, figure out what your elements are made out of and their bonding structure. Right? The last thing it tells you is how many bonds an element typically forms. Right? And the easy way to remember this is your ideal position. Elements want to be like these so-called noble gases, helium and neon and argon. And so in order to get there, they have to add electrons. Right? If fluorine or chlorine is going to look like a noble gas, it has to take on 1 electron. And so these elements tend to form 1 bond. Oxygen, sulfur, etc., they need to take on 2 electrons to get there. So they tend to form 2 bonds. Nitrogen forms 3, carbon forms 4, etc. these rules will be very helpful when you're looking at the chemistry of biological molecules, because it tells you how many bonds each atom is likely to form. That makes it a lot easier to follow chemical reactions when they're drawn out. Okay, so let's look at some examples. This is showing the formation of water, and it illustrates probably the most important type of bond that you're ever going to come across in chemistry, which are called covalent bonds. And a bond really just means that two atoms are connecting to each other in some way. And a covalent bond means that they share electrons. This oxygen, its outer shell can hold a maximum of 8 electrons, and hydrogens can hold a maximum of 2. Now you notice the oxygen only has 1, 2, 3, 4, 5, 6, and the hydrogen only has 1. Okay, so what we can do is we can have them share. If oxygen and hydrogen share 1 electron each, right, and we form 2 of those bonds, everyone's satisfied. Each hydrogen gets 2, that's what it wanted. The oxygen gets 8, 1, 2, 3, 4, 5, 6, 7, 8, that's what it wanted. And so covalent bonds form because, if you imagine that atoms have preferences, it's in everyone's best interest. The oxygen wants 8, the hydrogen wants 2. If they agree to share, everyone's satisfied, and they wind up looking like those noble gases with full electron shells that we see at the end of our periodic table that are the most stable elements out there. Okay, so covalent bonds, sharing of electrons, think strength and stability. These are the strongest bonds that can form. They're the most stable. So when you want to build complex molecules like DNA or protein or lipids that make up cells, these are by far the most common bonds that are used to make that happen. And they're just using water as an example, but this could happen in virtually any molecule. Okay. All right, if you want to join atoms, but there aren't enough to go around to use single bonds like we saw back here, in water, each of these is a single bond because each atom is sharing 1 electron. If you want to directly bind 2 oxygens to each other, well, they each start off with 6 electrons. We want them each to have 8 total. If you're going to make that happen, they're going to have to share 2 pairs of electrons. We designate that, show it by having 2 lines connecting them. That just means 2 electrons are being shared. For some atoms like carbon, triple bonds are even possible. They can share 3 sets of electrons. Okay, so you get to the same end result. They both get those 8 electrons that they need to make their electron shell seem full, but it's just 2 pairs that are being shared between the same set of atoms. Okay. All right, second type of bond. Ionic bonds are different than covalents in that instead of the electrons being shared between 2 atoms, it's a complete transfer. Okay, so in this example, sodium and chloride combine to make salt, right? This is the same exact table salt that you have in a salt shaker in your kitchen, and sodium has 1 extra electron. If it gets rid of it, it'll drop down to its previous shell, electron shell being the outermost one, and everything's good because that one's full. Chlorine is just 1 electron short of having its outermost shell being full. So if we want to make them both happy, sodium is more than willing to give away its electron, and chloride is willing to take it. They both wind up with full electron shells. The sodium winds up with a positive charge because we lost an electron, right? If you go down a negative, that means you're +1. The chlorine winds up as a minus charge because we added an electron. And so just like adding positive and negative numbers in math, if you lose an electron, you become positive. If you gain an electron, you become negative. And when we put that sodium chloride in water, they happily split apart into those separate positive and negatively charged ions because they interact with partial positive and negative charges on water molecules itself. All right. So that leads us into a slightly different concept involving polarity, which is describing the way that bonds take on partial positive and negative charges within one molecule. So I'm going to save this for the next video. We're going to talk about polarity, look at how it creates hydrogen bonding between water molecules. So follow along to the next one, and we'll keep the discussion going. Make sure you flag the playlist, subscribe to the channel. New videos will keep popping up as I work through the book.